The overall goal of this procedure is to induce migration in typically non-migratory cells to identify specific compounds that activate or block migration.
This method can help to answer key questions in the field of cell migration and invasion and may be of particular interest to scientists working with neural stem cells.
The main advantage of this technique is that non-migratory and non-invasive cells can be analyzed during their process of becoming migratory and invasive.
The implications of this technique extend toward the identification of novel compounds that block epithelial to mesenchymal transition, or EMT, which occurs in human high-grade gliomas believed to derive from neural stem cells. Though this method can provide insight into EMT in neural stem cells, it can also be used to study other epithelial cancers such as breast, lung, or colon cancer.
On embryonic day 14.5, confirm the correct age of the rat embryos by the presence of beginning digit formation in a rat forelimb. Next, transfer the embryos into individual uncoated petri dishes filled with ice-cold PBS, which are placed under a stereo microscope. Use a pair of fine-tipped forceps to remove the embryonic hulls. Keep embryos in expansion medium on ice. Using 2 autoclave sterilized fine forceps and starting at the intersection of the developing telencephalon and diencephalon, pull simultaneously anteriorly and posteriorly to remove the head.